Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this lecture, I will be going over biosynthesis of nutritionally non-essential amino acids. Let's get to see what are the nutritionally non-essential amino acids and how they are synthesized in our body. Now, it takes higher number of metabolic reactions and lot more ATPs and the energy that need to be spent to synthesize nutritionally essential amino acids, like synthesis of histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine. It takes lot more reactions and more energy, especially if other than arginine and histidine, all other nutritionally essential amino acids. So it makes sense in an evolutionary point of view, it makes sense to take them from the diet and synthesize the ones that takes less energy and needs less metabolic reactions. And nutritionally non-essential amino acids, they take less energy and also need less biological reactions to make them in our cells. So that's why evolutionarily, so synthesis of essential amino acids have been kind of taken out of the system and made our body to be dependent on them from the diet coming from the plants and evolved to synthesize only non-essential amino acids, and that's what is the rationale here. Now, what are the nutritionally essential amino acids? Arginine and histidine are usually considered as a semi-essential amino acids, especially the arginine. Isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan and valine, which can be remembered as TV till PM. That's a mnemonic. TV till PM can be used as a mnemonic. Let me write it down. To remember a nutritionally essential amino acid, you can use TV till PM. This gives all the names of essential amino acids. T for tryptophan, V for valine, another T for threonine, I for isoleucine, L for leucine, another L for lysine, P for phenylalanine and M for methionine. So other than these, other two like arginine and histidine are considered as semi-essential amino acids because they are necessary only under a positive nitrogen balance condition. Coming to nutritionally non-essential amino acids, other than these that I have mentioned, like 10 of them, especially 8 absolutely essential amino acids, all others are referred as nutritionally non-essential amino acids. Now let's get into synthesis of each one of these nutritionally non-essential amino acids. The first one is to synthesize glutamate and glutamine. Now the cells are going to use alpha-ketoglutarate. And the alpha-ketoglutarate is a TCA cycle intermediate, so the cells can get that from glycolytic reactions like glucose to pyruvate, pyruvate to acetyl-CoA, acetyl-CoA going into TCA cycle give alpha-ketoglutarate. Alpha-ketoglutarate is used to fix ammonium ion done by glutamate dehydrogenase reaction to make glutamate. That's how non-essential amino acid glutamate is synthesized. So the synthesis is done by glutamate dehydrogenase enzyme by fixing the ammonium ion to alpha-ketoglutarate. Now the glutamate is going to be fixed with one more ammonium ion in the side chain, that is called amidation process. Side chain nitrogen is added there, amino group, side chain amino group or amide nitrogen. And that's how glutamine is synthesized. So done by glutamine synthetase enzyme, which needs ATP to do this. So this is how glutamate and glutamine, two amino acids are synthesized, that's why these are non-essential. Means we don't need to take them in our diet. Only two reactions In fact, 1-1 reaction for each amino acid. Coming to synthesis of alanine. Alanine is synthesized by transamination reaction. So in the transamination, alanine transaminase is the enzyme which needs pyridoxal phosphate, and using this pyridoxal phosphate as a transfer intermediate for amino group coming from glutamine, which will be transferred to a pyruvate, which is a keto acid coming from glucose oxidation. So the pyruvate is converted to alanine after it receives amino group from glutamate, done by transamination reaction catalyzed by alanine transaminase. That's how alanine is synthesized. Now let's move on to see how aspartate is synthesized. Synthesis of aspartate also involves transamination reaction, like how we have seen with the synthesis of alanine. So the transamination of oxaloacetate, which is a keto acid here, which is an intermediate in the T-share cycle, and also oxaloacetate can be synthesized from pyruvate by pyruvate carboxylase enzyme. So once we get oxaloacetate receives amino group from glutamate, which is mediated by pyridoxal phosphate catalyzed by aspartate transaminase enzyme. And aspartate transaminase, previously called as 𝐒𝐧𝐫𝐞, and alanine transaminase in the previous slide, it was also known as 𝐻GPT. Now, this is how aspartate is synthesized from the transamination reaction. Once we get aspartate can undergo transamidation reaction, that is transamidation of aspartate, especially the side chain carbons of aspartate be added with amino group, that is what is referred as transamidation. Note the difference between transamidation and transamination. The difference is, in transamination, amino group is transferred to alpha carbon, whereas in transamidation, amino group is added to the side chain carbon. That's what is the difference in the naming. Remember that. So the transamidation of aspartate, it is done by asparagine synthetase enzyme. And the amino group donor in this transamidation of aspartate to make asparagine is a glutamine. Amino group donor is glutamine here, and the amino group of glutamine, side chain amino group of glutamine, in fact, will be transferred to aspartate to make asparagine, done by asparagine synthetase enzyme. Some of the tumor cells like acute lymphoblastic leukemia, referred as ALL, acute lymphoblastic leukemia. So this acute lymphoblastic leukemic cells, they are deficient in asparagine synthetase enzyme. So they are referred as asparagine synthetase deficient tumors. So there are some tumors which are deficient in asparagine synthetase enzyme, and ALL, acute lymphoblastic leukemia, is one such tumor. So how these tumor cells will proliferate, how they are going to get asparagine, which is essential for protein synthesis. So the tumor cells like acute lymphoblastic leukemia cells, they are going to get asparagine from the blood. How asparagine will be present in the blood? So whenever we take protein diet, so asparagine enters into our portal blood from the diet after the digestion and absorption of a protein. So, and that asparagine is there in the blood, and acute lymphoblastic leukemia cells will take that asparagine and proliferate. So this particular concept here, knowing that acute lymphoblastic leukemia cells are deficient in asparagine synthetase, they are heavily dependent on blood-borne asparagine. So there is a drug that has been designed which is an exogenous asparaginase enzyme, and this exogenous asparaginase is marketed as L-SPAR, that's a brand name. So what this exogenous asparaginase enzyme will do is, this asparaginase enzyme is going to convert asparagine in the blood into aspartate. That's what asparaginase will do. So by converting asparagine into aspartate, they are going to decrease the blood levels of asparagine, and thereby acute lymphoblastic leukemia cells, they won't be able to get sufficient asparagine from the blood. So basically, by providing exogenous asparaginase, so we are trying to decrease the available blood levels of asparagine for acute lymphoblastic leukemia cells, thereby targeting their protein synthesis. What about normal cells? Normal cells, we don't need to worry, because normal cells, they have their own asparagine synthetase, and they are going to make asparagine from aspartate. That's the concept that you must remember. Coming to synthesis of serine. Serine is synthesized by oxidation and dephosphorylation of glycolytic intermediate, that is 3-phosphoglycerate. 3-phosphoglycerate, as it is shown in the figure here, is oxidized into phosphohydroxypyruvate, and which undergoes transamination to become phosphoserine, and phosphoserine undergoes dephosphorylation process to become serine. So that's how 3-phosphoglycerate, that is a glycolytic intermediate, be converted into L-serine, which is a non-essential amino acid. That's why it is non-essential. We don't need to take that in the diet. Alright, now let's move on to see synthesis of glycine. Now the glycine can be synthesized from serine by serine hydroxymethyl transferase. So here in this reaction, a tetrahydrofolate will be converted into N5N10 methylene tetrahydrofolate, while serine is converted to glycine. So this is one way to synthesize glycine in our cells. Now the other way to synthesize glycine is a transamination reaction of glyoxylate in the peroxisome, where glyoxylate is transaminated with alanine to make glycine, and the reaction is catalyzed by enzyme alanine glyoxylate transaminase, which is simply written as AGT. Deficiency of AGT will give rise to a disease called primary hyperoxaluria. So in the primary hyperoxaluria, there will be increase in oxalate stones, which can lead to renal failure. And the third way of synthesizing glycine is conversion of choline into betaine, betaine into sarcosine, and sarcosine in the process will be converted to glycine. Now I have already explained you about primary hyperoxaluria, which is one of the clinical application here related to glycine. Coming to proline. Proline is synthesized upon conversion from glutamate. Glutamate is converted to glutamate semialdehyde, and glutamate semialdehyde further it will be converted into proline. So one of the way to synthesize proline in the cells is from glutamate. Coming to formation of cysteine. Cysteine, it is synthesized from methionine, because methionine is a sulfur containing amino acid. Cysteine is also a sulfur containing amino acid. So the sulfur which is present in cysteine, it is actually coming from a methionine. So the methionine here, it is simply a sulfur donor, because the carbons present in cysteine, they are all coming from serine. So serine, amino acid serine will donate carbons present in cysteine, and sulfur is taken from methionine. Now how that is done? Methionine, which is converted into homocysteine, and homocysteine, while it is converted into cystathionine, done by cystathionine beta synthase, and during this process serine enters into this reaction and going to give the carbon skeleton for cysteine formation, and the sulfur will come from methionine. Okay, so that means there are two enzymes that are involved here in the conversion of homocysteine into cysteine. One is cystathionine beta synthase. So here the transfer of sulfur present in homocysteine will be done to serine, thereby serine becomes cysteine. So that reaction needs cystathionase also. So with the help of cystathionine beta synthase and cystathionase enzyme, our cells will be able to synthesize cysteine. That's why cysteine is a non-essential amino acid. Remember, the carbons of cysteine are actually coming from serine and the sulfur in cysteine is coming from methionine. So as long as methionine is there, so our body will be able to synthesize cysteine. Coming to formation of tyrosine, which is another non-essential amino acid. Tyrosine is simply, it's an hydroxylated product of phenylalanine. Phenylalanine, which is an essential amino acid with its phenyl ring, will undergo hydroxylation process as it is shown in the figure here to make tyrosine. So the difference between phenylalanine and tyrosine, it is tyrosine has this hydroxyl group attached to the phenyl ring, making a phenol here. And the coenzyme for the conversion of phenylalanine into tyrosine is tetrahydrobiopterin, which in this process will be converted into dihydrobiopterin. That is BH4. BH4 is tetrahydrobiopterin. BH4 is converted into BH2. And later this BH2 will be converted back into BH4, and that will be requiring NADPH plus H plus molecule or NADH plus H plus molecule done by dihydropteridine reductase enzyme. So two enzymes are required here for the sufficient conversion of phenylalanine into tyrosine. One is phenylalanine hydroxylase, the coenzyme tetrahydrobiopterin, and also the recycling enzyme called dihydrobiopterin reductase. Dihydrobiopterin reductase is also necessary to make sure phenylalanine is sufficiently converted into tyrosine. If there is a deficiency of tetrahydrobiopterin, or if there is a decreased activity of phenylalanine hydroxylase, or if there is a decreased activity of dihydropteridine reductase, all of them will give rise to different types of phenylketonuria. Remember that. Now coming to formation of hydroxyproline and hydroxylysine. Hydroxyproline and hydroxylysines they are not a standard amino acids, meaning we don't get these amino acids coming from protein synthesis using a codon, but these are the ones that are part of our proteins, especially collagen, and they are synthesized upon post-translational modification of proline and lysine. So the proline and lysine undergoing post-translational modification done by prolyl hydroxylase and lysyl hydroxylase, which needs ascorbate. So they are made within the polypeptide chain itself. So that is the example for post-translational modification, and vitamin C ascorbate is the coenzyme here. And deficiency of vitamin C will lead to scurvy, where there will be decrease in the conversion of proline into hydroxyproline and lysine into hydroxylysine. So this is all about how our nutritionally non-essential amino acids are synthesized in our cells. Thank you.